All right, welcome back everyone from lunch. Uh, I have to be here again with you for just one more module, which will be 7.2, where I talk about dynamic compartments. The presentation and the files are being uploaded right now, and as soon as they're done, Heidi will share with you on the chat. But you can just keep going and follow along with me right now. So if you remember the my talk from Friday. Last week, we talked about the compartments in CompuCell3D, how that can be used to increase the spatial resolution of CompuCell3D or cellular plots model um, simulations. And we talk about two ways that we can go about it. We can either use compartments to represent structural uh, elements inside your cell. It can be the membrane, can be the nucleus, can be the Golgi apparatus, can be the mitochondria, can be the nuclei. So it can be very specific things inside your cell. Or you can also use the compartments to represent more abstract compartments at the cell. For example, if you are modeling an epithelium, then your compartments can be the apical side of the cell, the basal side of the cell that attached to the basal membrane, and the core of the cell that attached to or the lateral side of the cells that attach to other cells nearby. Okay, and uh, we have seen a few examples of the both uses. So on the left again is the use of the specific structural cell compartment to model crawling cells. And in this model, we actually model the nuclei of the cell, the lamellipore of the cell, and the rest of the body of the cell, which are where everything else it is. But you can also model the apical and basal compartment of the cell and to modern epithelium, like you see in this example here in this movie, where you see the same thing using 3D. So I know that you have not done many 3D simulations in this workshop because we want to keep the examples simple and fast, but everything that you're learning here can be done in 3D. Uh, and below here, you see an example on epithelium. And on this example, I highlighted the FPP links that are connected to the apical side of the cells. And in the next session that will be presented by Pion Adiapak, you'll be uh, adding this kind of FP links between the cell compartments and make them change over time to make the cells undergo apical constriction and bend the tissue. But all this is very good, it's very powerful. So if you combine cell compartments with FPP links, either internally or external FPP links, then you can do a lot of things. It's very powerful. But I would like to, for this, but there are some kind of shortcomings that when you use cell compartments, okay? Even when you use abstract cell compartments, uh, one of the limitations that you still have is first that usually you put the compartments at fixed place inside the cell, so they have fixed positions. For example, the nuclei in the center, the lamellipod is at the edge, and you have to choose where you're gonna draw your lamellipod to begin with. So you usually put on a specific side to the right side of the cell or the top of the cell. Okay. Uh, and the other limitation is that if for some reason you decide to flip your epithelium from upside down, okay, the cells will have a very fixed time to adapt their positions. They have to repolarize in a different way. And if you use just the cell compartments as they are, it's going to take a very long time for your cell to actually flip because as one flip, all the other cells on the side have to flip as well. So that can be very hard and maybe not possible at all, depending on your simulation parameters. Okay. The other disadvantage is that, as I said, you must, because you have fixed cell positions, you have to be very careful about where you put your cells at the beginning of simulation, because if you put again on the wrong side, the cell will never be able to kind of remove their cell compartments to correct positions, right? And the other thing is that when you use cluster mitosis that already take care of the division of the cell into compartments, they recopy the compartments, you usually make exact copy of the where the compartments are. So if they have apical on the top, basal on the bottom, and then you divide the cells in half, the two other copies of the cell, you have the same arrangement of apical and basal sites. And that might be good for some applications and might be very bad for other applications, because in reality, when a epithelial cell divides, what it does is actually, if first lose its polarity, it rounds up, divide, and then regain its polarity and should repolarize back again after division. And that is not possible at all if you're just using static compartments as have been doing so far. Okay, so to overcome these limitations, we introduced the idea of dynamic cell compartments technique. Okay, so the idea here is that the following is that we do not pre-specify where the compartments are. We just put them randomly inside the cell and let the external contact energy of the cell dictate where those compartments should sort out to be. Okay. So the advantage of that is that there is no need to specify positions. The cells can quickly adapt to the changing environment. And when you do cluster mitosis, you first depolarize the cell, okay, add the division, and then let the compartments resort themselves according to the new position. So if you divide, if you have a cell that's compartmentalized along the epithelium, sometimes they have to divide on the x of the epithelium, sometimes perpendicular to the epithelium. It doesn't matter which x of division you choose, the cell should be able to quickly adapt and find their way back into epithelium. Okay, so how this actually works is by using you don't need to do any kind of hybrid simulation. We don't need to invoke something else from outside the CompuCell3D or even the cellular pods model. Everything is done within the cellular pods mechanism. Okay, so the trick is the following: is just when you have the contact internal energies, instead of just setting a low value, you actually go beyond that and you set a negative internal contact energies for all your compartments. So because when you do that, what happens is that you induce the cells to be in a mixed state. So all those cell compartments that you had before will now be, because they have negative energies if all the other internal cell compartments, they would like to mix together inside the cell. So you start by using your cell contact internal energy, but to induce a mixed state inside the cell. Okay, so uh, that seems at first that that cannot lead to anything good out of it, okay? But if you combine this internal mixed state of the cells that you should have otherwise with a appropriate choice of the external contact energies, okay, then what happens that if the cell is by itself, okay, then all the compartments are sorted inside. So, which also means that all the blue compartment is simultaneous touching all the other sides of the cell, and the same for the red compartment, the same for the green compartment, the same for the white compartment here. So they are simultaneous at all the cell surfaces and sensing what's going on cell surface. Now you've combined that with a positive external contact energy, which can guide where those compartments should be based on their immediate environment. So, for example, if you put this cell next to another cell, and this green compartment represents the adhesion molecules, okay, what will happen is that as soon as a green compartment from one cell sees a green compartment from the other cell, they kind of lock uh, themselves along the compartment, along that surface, and then the other green cells start kind of diffusing and going there, or that compartment grows by the intrinsic mechanism of, let's say, copy attempts within intrinsic to the CompuCell3D or cellular pods mechanism. And then you have an enrichment of the clinical parts that get enriched at the interface between those two cells. So you put the cells together, they already have an internal mixed compartments, but because by the self-interaction, they automatically go and co-localize at that cell interface without you having to do anything on it, okay? And you can also combine that to get more things. You can also have a cell compartment, for example, that likes to be in contact with the medium. You can think a medium about a buffer, or maybe the medium is a composed of ECM proteins, and your red compartment can represent the integrins inside your cell, so they automatically localize to the outside. So with this, uh, you can do lots of things. For example, one of the compartments can actually be the cytoplasm or the cell body of compartments that have a very high external contact energy with everything that's outside of it. So therefore, this white compartment localized to the center of the cell. Okay, and then again, maybe the red cells becomes the integrins, and your medium is uh, coarse grain representation of the ECM surrounding the cell, so they're gonna automatically sort out. Maybe your green compartment can be represented the cadherins. So as soon as two cells come together, they co-localize at the interface of the cells. Or maybe your compartments may be the green and blue compartment can be F and ephrins, transmembrane proteins that kind of localize in a heterotypic way. So ephrins do not like to contact other ephrins on the other cell membrane. They like to contact Fs and vice versa. So if you can choose your external contact energy such a way that ephrins and Fs, or the green and blue compartments, they have a very low contact energy between them across cells, across clusters. Therefore, you make a heterotypic colocalization of the blue and green compartments according to cell interface. So see here you have green here and blue on the other side, but below here you have reverse. You have blue on the cell on the left and green on the right. If you wait long enough, maybe everything's going to be green on one side and everything's going to be blue on the other side, or you place just another cell here, and then maybe the cell in the middle have all the green on one side and all the blue on the other side. Okay, so that is the idea here, and I'm going to show on this module here how we can work this out and how to choose those energies, and I give you a few examples how you can use those. Okay. Uh, is this making sense so far? Let me know if you have any questions before I proceed. Okay, so a few examples in the literature where this technique has been used. One of them was to use the a model, uh, a function and growing renal tube, where you have those cells here that they are polarized. Okay, so they have I'm using here dynamic cell compartments that stay kind of more or less mixed inside the cell when the cell is alone. And as soon as one of those cells that make the epithelium grow and divides is lose polarity, I replace the cell back the two other cells of the epithelium, and they automatically sort out the compartments to make the epithelium, right? And they have a little bit more rules to tell the cells to actually go and co-localize their places. So here's a video where you see uh, renal cells growing and forming a cyst. So you see that it starts in a pixelized state. I'm gonna show you how you can do that on the Python level. And then it sorts out as soon as you grow and divides, the cells start to co-localize their compartments that represent cell-cell adhesion. And then this grows up to make a cyst. So now you have the cells growing, dividing. Red's the basal membrane, green is the dark red is the basal side, green, light green is the apical side, and bright red is the cell-cell adhesion. So I'll just play that again. So here you see the same simulation, but here in 3D, and here's a cross section. And then you see the use of the dynamic cell compartments technique for in this paper. Okay, so another example here is a model. Here I'm modeling two things. First, I'm modeling mesenchymal-epithelial transition. So all those cells in blue, they're in mesenchymal state, and the light green here is some kind of luminal fluid. Okay, and at some point the cells become compartmentalized. It happens too fast in this simulation. So they have now light green, which is apical, red, that's basal, and the blue is just a core compartment. So they automatically segregate apical, blue to blue, and red to red, or red to the outside. And you also have internal cell FP links that makes the cells elongate. And over time, you form the structure. And every now and then, when a cell is not in a happy state, it completely loses polarity and repolarize again, and you allow the cell to actually self-correct this position. So that's what you see, this kind of things exploding here is just a depolarization, repolarization process happening. It's just that the video is too fast for you to really see. Okay, and a final example where this can be used is this model of planar-cell-polarity, where again the cells all start with the mixed state, but you have this kind of proximal-distal compartments that they like to colocalize across the cell surface. So where you have a green, you like to have a red, and vice versa. So over time, the cells polarize, and then over time, the whole tissue polarize, and all the cells have the same orientation across the tissue. So um, if you see here at the beginning, they have different directions, but as time goes by, eventually the whole tissue agrees to have all the red compartments to the left and all the green compartments to the right, or actually it's vice versa. They yeah, all the red to the left and all the green to the right. Okay, does it make sense? Do you guys have any questions before we proceed to the first exercise? So everything is good. Let's proceed to our first exercise. So in this first exercise, actually, it's going to be a series of two exercises. We'll be trying to reproduce these panels that you see here in the screen. What you see on A and C, also in B and D. Okay, so our objective is to recreate this image you see on the left panels of the figures. So for this exercise, the first thing I want you to do is to download the simulation template 7.2.1.zip, and I just want to check what is written in there. Okay, so I'll give you a few seconds for you to download it and open it under Tweeted. All right, so if you have it open on Tweeted, um, this is what you should see. Okay, first we can look at the XML file and see that we have four cell types defined: C, B, R, and G. So C will be the cytoplasm. Then we have blue, red, and green. And we also have a plugin called PixelTracker that is used so you can actually grab a cell and have information about where all the lattice sizes that belong to the cell are. And yes, Maisha, if you run simulation, you should get this kind of beach ball like output. That's exactly what we're doing there. We're gonna now modify this base file to get what we want. Okay, because and the reason you get this beach ball is because right now all the external energies are the same, Martin, and all the internal energies, except the ones between same cell compartments, they are two. Okay, so C and B, C and R, CG, CG, and all the compartments are positive. So they're not negative as I ask you to do. Okay, and of course, CC, BB, RR, and G, they are just zero because we have only one copy of cell compartment inside your cluster, so you don't need to define them. Okay. So what else? Okay, so because of this, no pattern should occur. And you get exactly what Maisha has shared with all of us in the chat. So let's continue. And then if you look now, it's still in the XML on the uniform initializer, you see that I created is my coordinates from 2020 to 4444 on the X and Y. That seems that I should put a lot of cells inside, but actually our cells is 24 by 24. Okay, that's the width of our cell. And so we are just drawing one cell in simulation. It's going to be it's a big cell, and it's of type C. Okay, so you should only have you're basically drawing a square of a yellow cell in the center. Okay, but if you run simulation, you can see that you get the outer compartments. How do you get the outer compartments? Okay, now we have to go to the Python level. So if you go to the Python steppables and look at the start function there, then you see this piece of code. Okay, so what this piece of code is doing? So first is going over all your cells of type C. Okay, so it's going all the cells of type C, which stands for cytoplasm in this case. Okay, and what it's doing is first is using this code here to create a cell of type B, R, and G. Okay, so you're now creating those cells types. Okay, so you have this comment self dot new cell, and then you have to specify the cell type B, R, and G, and we're storing then this variable B, R, and G. Okay, so to keep things simple. Then when you create the cell, that's a completely new cell that exists by its own, and it has its own cluster ID. So because we want to put that in the same cell cluster, the first thing you do, you reassign the cluster ID of the cell to be the same as the cell that you're looping over. So now they all are part of the same cell. Okay. However, if you just do that, you see nothing different in the screen because you created that cell B, R, and G, but you did not place anywhere in your lattice. So the next steps here is actually where you draw those cells inside your C compartment, your C cell. So what it's doing here is first on this line here, you're retrieving the list of all the pixels that belong to the yellow cell that you have at the beginning. So you get the list of all the pixels or all the lattice sites that these cells, that the cell occupy, okay? After you do that, you're going over all those pixels, okay? And then you're getting the X and Y position of that pixel. So that's just what those two lines are doing. And then for each one of those pixels, you're gonna flip a number between 1 and 6, okay? And if this number happens to be 1, you assign that pixel the color blue. If that number that you get out of it is 2, you assign that pixels the color red. If that pixels is of type if you get the number 3 out of your random event, then you assign that of type G to be green. And if you randomly check 4, 5, or 6, then you just leave it as it is. So you leave it as a yellow cell. Okay. So yeah, basically I'm going over all the pixels that belong to the cell and I make them red, green, or blue just by chance. And I make it in such a way that half of them will not be changed, but the other half of them will be changed such a way that you have 1/6 of them being blue, 1/6 of them being red, and 1/6 of them being green. Okay, questions? That's not a dumb question. That's a very good one. How do I know whether you want to put this under start versus initial condition? Init. The init function in your steppables is just a housekeeping function to initialize your steppable or your class, okay? So that's something that goes internally in CompuCell3D. The start one is actually that is called, once everything's set up, that is the functions that you're gonna be, all the comments that you'll be doing at the Monte Carlo step 0. Before you do any lattice side flip, that's where you put all your commands. That's a very good question. Okay, so it's good that we clarified this now. Yeah, the init function is just a housekeeping to initialize the class. The start is actually where you want to put all your code. Very good question by Robert. So in this case, yeah, the big compartment is going to be all over. Yes, and the same thing is true for the red compartment and the green compartment. They're going to be all over the cell. All those lattice sites are completely disconnected, and actually they may remain disconnected, especially if you choose a negative internal energy. So that's completely okay. It doesn't, the cells, compartments, they don't have to be continuous in the large site for it to work. Okay, you can still satisfy the volume constraint if you have all your pixels uh, pulverized lattice, right? As long as you have uh, uh, 50 pixels, then everything is okay. Okay, and how the substrate deal? It just do just fine. Okay, what tells all these different lattice sites that belong to the same cell to be together is actually the external energies, right? They do that because usually have positive contact energy with everything else, so they unite to cluster them together, and they remain clustered because you have positive energies. Make sense? Yes, yes, we have the case of the rendering lamellipolar that just got disconnected. In that case, you have a whole compartment disconnected, right? And then go back to the cluster. Okay, so the clusters, the compartments, they like to be together because they usually have lower energy than everything else. So they usually like to, so that's the trick to keep them together. Okay. Another question here. Very good question by Nehar. If I like, let's say the blue and the red part, they should be together because one is the endoplasmic reticulum and the other is the cell membrane, right? So in that case, you assign a even lower energy, internal contact energy between those two. So if they all have an internal contact energy that is negative, let's say -1, but those two compartments, they actually have to be together with one another, then you assign then internal energy to be -5, okay? Um, so you just play with the internal compartment energy, the relative energy of everything else, and then you make sure that those two actually stay together. So you don't really code it, you just make a wise choice of your internal compartment energies, okay? All right. So so this next set of exercise is for you to get familiar exactly how you chose those. Okay, we will not have a membrane endoplasmic reticulum example, but we have examples that maybe look like this. Okay, all right. So now you define the blue, red, and green cells. You make sure that they belong to the same cluster. You run the sign uh, then of pixelize. So you're pixelizing your cell into compartments. Okay, and after you do that, you cannot forget to assign cell volumes to all these new compartments. Otherwise, you're going to disappear in the next steps, right? So what you do, I don't try to guess how many cell lattice was assigned to blue, green, and red. I just make the blue, red, and green target volume to be whatever volume they end up with. Okay. And then I just assign a lambda volume, of course, they need to be constrained. And the same thing I do for the original cell that was C, right? Whatever pixels are left for it, that's going to be its new target volume. Does it make sense? If it makes sense, I want you to now to just play the simulation from Tweetit and tell me what you get. So probably you should get something that is very similar to what Maisha has. You should, when you just play the simulation, you should get a beach ball. Okay. So let me know if you get this output. And of course, exactly where the blue, the red, and the green compartments will be is purely by chance, right? Yeah, and you can see that everyone got a different configuration. Uh, some of them get Kyle got the green detached from the blue and the red, which was nice. Uh, yeah, so the exact position of them is going to be completely random, right? So no one has the same output except for Aritra. Aritra got the same beach ball that I got, but rotated 9 degrees. which was also a nice output. All right. So now that we got this, the question is why did it not remain mixed inside? And the reason it did not remain mixed inside is that uh, they all have still positive contact energies inside. They are all two, right? Because they're all two, it's better for them to have to be together with themselves because by definition, a red and red pixel have zero energy between them. Yellow pixel has zero energy between, and a green pixel has zero energy between them. So that's just a definition of that's the definition from the cellular pots model, but zero is uh, the neutral energy is defined as zero, right? So that's why your cluster likes to be together. So to overcome that and induce a mixed state, you have to make all those energies to be negative. So your next task here is let's induce mixing of the cells. So try to do that and let me know if you get the cells to look like this. Okay, Hago got it. Our -2, so then we got a mixed state. Red, green, and blue, you actually don't need to specify. So actually it's kind of grayed out here because you only have one cell compartment copy inside each cell. Ildes also got it. What I want you to notice here is that if you just set it to be zero, okay, you would think that should be enough to induce a mixed state inside the cell, but it's not because of the way that the domains grow inside the cellular pots model. If you just make zero, you have a little bit of mixing because green, blue, and blue, all the other colors are the same. But remember that a lattice site it can only grow by copying one to the neighboring pixel. So because of that, there is still some kind of um, pixel to pixel correlation, spatial correlation between them. So to really overcome that correlation that's intrinsic to the way that the lattice sites are updated in the cellular pots model, you have to actually force that a little bit by making um, the energies to be negative. So for me, when I choose -5, I got a really good mixing. Okay, but as you have all been showing, negative energy of -2 is already good enough to induce that. Okay, so excellent. Very happy to see all those screenshots. Okay, thank you, Aritra and Leonardo, and also Nathaniel. Now the next challenge for you now is I don't want to, let's say that my yellow is not so what those colors means here, okay? What they exactly mean, it really depends on your model. But I usually like to think about these mixed compartments, the dynamic compartments, as a set of proteins, okay? They have different properties and they like to localize the same space on the cell, okay? So you can think of the green as cadherins, or the red as integrins, okay? Things like that. It can be whatever group of proteins. So it's something that can diffuse inside the cell or diffuse along the membrane, in which were under without any external input, they're just going to remain diffused inside the cell. And once that something comes up, they segregate. But one of those compartments can also be something generic, like it can be the cytoplasm, like I have in the example before. So let's say that our yellow part should be the cytoplasm, and everything else that's colored should be proteins that sits on the membrane. Okay, so it should be something like that, kind of looks like this. So the next task to you is make the cytoplasm to sit inside the cell. Okay, try to do that. That's a challenge now. Okay, Maisha, show how it happens if you stop halfway from change 2 to -2. Thank you. Okay, but that's a challenge to all of you. Try to make this yellow compartment to be inside the cell. And I don't want to be on the side, I want to be exactly in the middle. Okay, that's a challenge. And if you want to do this, what you should change? Which uh, energies you should change in your model? Should you change something in internal contact energy, the external contact energy, or on both? So I can see that Yudis got it work. So Yudis, what did you do to get this result? Ah, change contact for cytoplasm and medium to a huge number and cyto to other internals to be a little bit higher than cyto-cyto. So Adita also got it. Okay, excellent. So this was a little bit more. And Hago also got it. Excellent. Very happy. So this was a little bit more challenging one because you actually have to change two things at the same time. Uh, you have to change because you want the cytoplasm to be in the center, right? What you should be changing is the energy of the cytoplasm or the yellow compartment to everything that sits outside, okay? Or maybe towards the medium. So what you want to is to go out, actually to go to the external energies and change the cytoplasm contact energy with the medium to be a very high number, like 40. This will ensure that the cytoplasm never touches the cell membrane, it actually stays inside. But if you only do that, you get the cytoplasm to be inside, but all the other compartments are still happy to mix inside the cytoplasm. So you still have a lot of those other compartments that goes inside. And maybe it's this something that you really want to happen, okay? So that's not a wrong output. It really depends on your objective of your model system. But if you want that have all these internal auto compartments, red, green, and blue, to actually sit on the cell membrane, then you have to go back to the internal contact energies and set all the contact energies of the cytoplasm and blue, red, and green to be slightly more positive. And once you do that, then you get all of them to be outside. But you only want to do that between cytoplasm blue, cytoplasm green, and cytoplasm red. Because if you also do that with the other cross-section terms, you're going to get something that looks more like HANA has, where the green, red, and blue also mix out. So HANA, you probably made the green, red, and blue energy between themselves internally to be positive. Am I right or wrong? So again, you just make this with the cytoplasm internally and leave all the others negative, and then so if you make red, green, red, and blue, red, and blue, green to be negative, then you get back this mixed state that you see here on the panel on the right. Okay, question from Aisha. Just for my structural understanding, the types of energy pairings are repeated in both contact and contact internal. Yes, most of them. For example, in the contact internal, you don't need to specify the medium, right? So that can be excluded. Is it correct that the contact section is in interaction between all compartment cells, while contact internal is just for the members of the same cluster number? Yes, exactly. You got just right. The internal contact energy is just for the compartments that belong to the same cluster. And while contact is actually the external, so it's between compartments that belongs to different clusters. Yes. Okay, now do you guys have more questions? If not, let's proceed to the third task. Uh, Now try to get the red compartments to sort to the outside. So let's say that the right compartment really like the medium because it's something that attached to whatever the medium represents, and you want to get them to go outside. So let's try to do this. And you just already got it. Okay. So what do you have to change to get this output? Reduce contact red and medium. Okay. Yes, they are medium contact to something very low. How much low should it be? Should it be negative? Should it be zero, or should it still be positive but low? Yeah, if you make it negative, if you make like, especially if you make it very negative, like -10, what happens that you're gonna start to, then you just disperse into the medium, right? So you want to be low, but probably not negative. If you make negative, then you start to be the output, the contour of the cell become very rough. Maybe that's something you need. So you can try to make it -1, for example, or maybe -2 can still get away. But if you drive a little bit more than that, then you probably the right compartment is gonna pulverize and go to the medium. Okay, excellent. Levi got it. Okay, Maisha got it. Okay, so all of you are getting it. So are you getting more comfortable with uh, this idea? Excellent. Leonardo tried make the red and medium external, of course, to be -5, and then they start to break out. Okay, probably if you do something more like -2, you're gonna be a little bit better. You get the roughness, but not the dispersion. Okay. Neher got something that's completely well. Neher, you did got the red to go the outside, but the you probably want to make the internal energy of red and blue to be -5, so then they'll become mixed. So Neher, if you want to, okay, yeah. So make the red and blue internal energy to be -5, and then you should get something that looks like I have in my screen. So if everybody's good, let's proceed to the second part of this exercise. Let's now try to reproduce what happens when we have two cells interacting, okay? Uh, and let's try to produce this kind of cadherin co-expression, or maybe the F and ephrins co-expression compartments. So to do this one now, what you have to do, you have to actually draw two cells. And now you may be worried about how you're gonna do that in the code. But it's actually very simple. You already have the a start function that pixelize how many cells you have there. So you don't have to repeat that code because I already go over all the cytoplasmic cells, the C cells. So all you have to do, you actually draw by hand a condition such a way that you actually have two cells. Okay. So you all have to do is to go to the XML file, and instead of having your coordinates to be, for example, X from 20 to 44, you just add more 24 pixels to it and make your X go from 20 to 68, because then you are drawing this rectangle, and in this rectangle you can actually fit two cells inside. So if you just change that, okay, you should get two cells at the beginning that are, then the start function will take care of pixelizing the cell for you, but then you should get something that looks like this, okay? So let me know if you got that. So go to XML, change that line of code from 44 to 68, and tell me if you get this out. So you may see that it's kind of very hard to see the line that divides the compartments. So what I like to do when I play with cell compartments is I go to the visualization on the CompuCell player, right? And I unselect the cell borders, and I select the cell cluster borders. Okay, so you do not see there is no boundary being drawn between the cell compartments that belong to the same cluster, but there is a boundary that's drawn between cell clusters. Okay, and the other thing that I would like to do is when you click on the tools inside the player, you can select the cell cluster border to be black. So that's what I do in these examples here. So I only have the cell borders being drawn now, the cluster borders being drawn out the cell borders, and I make my cluster border color to be black. Does it make sense what I'm saying? So I just gave you tips for the visualization. Okay. But it's anyway, it's good to have your cell borders to be a different color from the cluster borders. So then you can really see the outline of what makes a cluster and what makes just a cell compartment inside a cluster. Okay. Now, the next task for you is to try to keep the cytoplasm compartment to be inside the cell and not to co-localize on the cell interface. Okay. So the reason that they co-localize is because we probably just changed the content of cytoplasm, the medium, the external medium to be very high. So the cytoplasm does not like to be with the external medium, which on a cell by itself makes the cytoplasm go inside because the cell does not see other cells. But now that the cells see other cells, the cytoplasm is completely happy to share contact with another cytoplasm from the opposite cell. Okay, so that's why they kind of co-localize like this. So if you want to make cytoplasm to actually be always inside the cell, what you have to do, you have to change all the external energies of the cytoplasm to be very high. So therefore, it does not like the medium. It does not like this blue cell from the other cell. Does not like the red from the other cell, and does not like the blue from the other cell. In other words, it doesn't like anyone that's from outside its cluster. Okay. So think about the person that hates everybody. So they always stay inside their house. So that's the behavior that you have to induce for your cytoplasm. So in this example here, I just make in the external contact energy, make a cytoplasm with everything else to be 40. Okay. Did you get this output? Okay, excellent. So you got this. Okay, so now your final task for this exercise is now to reproduce B and D. Okay, so don't need to do both, just choose one of them and try to get this out or that out. Maisha almost got the D. You have to make sure, Maisha, that if this is what you're trying to produce, that the green localizes with blue and vice versa, and not green green, no blue blue. Iris, you almost got it. That's a little bit noisy. Okay, try to make sure that you only have blue and green across interfaces. You still have lots of green green, and you still have lots of green and green. Okay, Kyle got it. Part D, excellent. It looks very nice, I'll say. The cells try to be separated, but the green and blue, they keep them glued together. Aritra, I think you're trying to produce B. Make sure that the cells only contact each other through the green and green. Okay, Levi got it. That part. You can also make a mixture. You can have the D, but only green green inside. So it depends on exactly how you chose those external energies, what the kind of cell coupling you wanted to have. You get different outputs. You can even have something that preferentially internally attached to the red. So now you already have the red that like goes outside. You can think of the red compartment in D as being the membrane. And maybe you want your green cell to be with the membrane. Wherever the membrane goes, the green cell goes. Okay. So you can also try that. So you should have a layer of red followed by a layer of grain. Okay. So to be the endoplasmic reticulum example that your colleague said before. Excellent, Hannah. Very happy to see that you got the B. Okay. Okay, and how good got a mixture of the B and the D. Okay. So do you guys have any questions? I'm gonna just place here my solutions for this exercise, what I have changed, and what values did I use to get each one of those outputs. So if you don't have any questions, let's proceed to the next exercise. Okay. And so this is very good. The exercise that here is you have the compartment that they automatically sort out. You can even try to modify the initial conditions to have three compartments or four compartments or more, and the same principle should work out. And then depending on your choice external energies, you get different kind of sorting out between the compartments. And that's nice to create a epithelium and let the compartments sort out to their places. Okay. Uh, but you can also use the same technique to actually induce things like to model epithelial and mesenchymal transitions between cells. So you can kind of polarize your cell, add out compartments, and the cell becomes polarized and the form epithelium. But maybe you want to depolarize your cells, okay? And they should go back to another state where they become mesenchymal. And actually, you can loop through these states, and that's what we'll be doing in this exercise next. So we'll be doing something that looks like you have seen here. And by the way, this exercise is based on a project that my student Abisha has been doing that will come out in a paper later. But let me just kind of remind you about what epithelial versus mesenchymal cells are, okay? So there are two different states that the cells can be, generic states. The cells can either be inside an epithelium, and if they inside an epithelium, they are usually forming a barrier or a structure, usually a two-dimensional structure, and the cells have to have a tight coupling between each other. So they differentiate between apical, have an apical part, a basal part, uh, or the cells can also be encountered in a mesenchymal state where they kind of separate with each other. They don't have a clear polarity, at least not an apical basal polarity, and they just kind of move. They usually more motile and move by themselves. Um, so they when the cells spread out, they basically do not form a tight epithelium. Okay, and there's the cells. They can transition between those two states, right? When you go from the epithelial to mesenchymal, it's going to be an EMT transition, epithelial to mesenchymal transition. And they also go the reverse state, going to be mesenchymal to epithelium, so MET, okay? And this is just a list of what the cell structures usually look like. Uh, but one thing that's important to say is that uh, it's not a black or white shade between what's an epithelial cell and what's a mesenchymal cell, okay? It's more like a gradient. The cell can acquire more properties that make it more epithelium, or can acquire more properties that makes it more mesenchymal. So it's a gray scale between an epithelial state and a mesenchymal state, okay? Uh, so some cells are more epithelial than the others, some cells are more mesenchymal than others, okay? And it can be more mesenchymal in different aspects and more epithelial in different aspects, okay? And they can transition between those two, okay? So during development, they do have the transitions, okay? So you can have differentiation where you have a epithelium of one kind and becomes a different epithelium from the other kind. But you can also have different type of epithelial mesenchymal transitions. For example, you have a primitive epithelia where those endothelial cells undergo um, uh, become mesenchyme, and then they maybe migrate to a different part of the cell, and then they repolarize again and form a secondary epithelia, or maybe your secondary epithelia can later uh, undergo transition again and become fibroblasts, or maybe, and also you have this kind of transition happening during cancer, and that's where a lot of effort in modeling and actually has gone into is to understand when you have this epithelial mesenchymal transition. People usually are focused on this first part here when you go from an epithelium that is sick and that induce metastatic thermal cells. So EMT are very high topic of research. But I'll say that equally important than EMT in cancer is also the reverse when your metastatic cancer cells become back into an epithelium, because it's actually this transition here that actually kills you, because uh, the problem is not this cancer cell spreading, but it's spreading and then form another cluster of cancer cells in other parts of our body. So I'll say that both of them are important. Of course, if you kill this first part here, you get rid of the second part. You don't have to worry about the second problem. But this second transition is also just as important as it is. And from what I see in the literature, there are very few people actually studying the second part here in cancer contexts. But I may be completely not up to date to the literature so far. So I already said that it's a hallmark cancer and uh, it's a spectrum. Okay. All right. So that's my very crash course introduction to epithelial mesenchymal transitions, right? So what you want to do now, you want to make a model that's going to be, we're actually going to do these transitions uh, back and forth. So I'll ask you all to please go ahead and download file 7.2.3 zip, and I want to open and take a look at what it is inside. So please download that. Yeah, James also making the case that epithelial tissue is also crucial in development, homeostasis, and wound healing. Right, you have epithelium and you make a wound there. The cells around the wound they have to go EMT to migrate to and also divide and fill the wound and then go back to an epithelium. Yes, Misha, you are exactly correct. The XML does not initialize the cell. That's done on another part. So I'm gonna go to that. So if you already open the file, uh, you see that it's a little bit similar to what they had before. Here we have four compartments, C, A, B, and L. C would stand for the cytoplasm. And now we have A and B, which stands for apical and basal. And you also have the L that stands for lateral. And you also have another cell type that's called basal membrane. Okay. All the cell starts as C and gets pixelated later, like we had before. The internal and external energies had already been chosen for you, so you don't have to work about that. That's going to be the next exercise. And the cells undergo EMT at the given time, and after some time they go MET, and then MET, and then they keep circling back between those two, polarization, depolarization. That's a way that I like to do, because then I can create just a single layer measure of VM and not have my wall to be of multiple cells. Okay, so let's go through the. I think there's no surprise on the XML part of it, but let's go through the Python level. So as Misha has seen, uh, the cell drawing happens at the Python level. So let's take a look at that. So if you go to the class init condition, the init function is again just housekeeping. And the start function, I have a loop to create four cells side by side, and they are all of the type C. And I just kind of change my ref X not reference, going to be this point here, this point here, this point here, and this point here. So this loop is doing exactly that's going here, 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 here. And after I get this coordinate, then I just make a cell that go from here, 12 on the side, 12 to the side, and I make this kind of box. And it's all of C cells, the same thing for this one, for this one, for this one. And then on these lines here, I just create a basal membrane that extends from here, oops, all the way here. And I just make the lambda volume to be very high. I set it equal to the current volume that I draw. And I make very high so it stays a rigid wall. Okay, so you see that I create initially a cell of type C. Okay, that's it. And then what I do later, I call this function called pixelate. That's going to do exactly what we did before, where you just place all the apical, basal, and lateral compartments inside the cell in a runaway. Okay, does make sense? So first I create all the cells as yellow, and then I add the pixels inside in a random fashion. Okay, so let's go to this pixelation function. So that you see on line 47. And the structure is very similar to what you had before, almost identical to what you had in the previous exercise. Okay, I first create a A, B, and a lateral cell. I assign them the same cluster ID. Then I go over all the pixels of the cell I'm pixelating. And I again make a random choice. If it's one, I make A cell. If it's two, I make a B cell. Now, this time I want my lateral cell to be actually double the volume of the apical and basal cell. So if my random number is either 3 or 4, I assign this a lateral cell type. And also I do, and everything else that is kind of either 5 or 6, I let it be cytoplasm cell. And and after I do that, I just assign the cell volumes, target volume to actually the volume that it has. So basically what this pixelating here is doing is I'm making this kind of all yellow cells to be pixelated like this. Uh, any question about that? Everything's okay? Does it make sense? If it does, please type yes, makes sense. If it doesn't, please unmute yourself and let me know if you have any questions. Okay, thank you. Makes sense. Also makes sense to Hannah. So I trust that that's good. So if you do that, you have all these cell pixels, and they're gonna sort out according to the contact energy that you had before. And I set the contact energy such a way that apical cell compartments like to go to the top, base compartments like to touch the basal membrane, and the lateral cells like to touch all the lateral cells, and the yellow cell, the C cell, like to be inside. Okay, but what's cool about this model is what happens on the step function. Because on the step function, I'm checking that every 2000 Monte Carlo steps, I either go into a mesenchymal to epithelial transition or the epithelial to mesenchymal transition. So I set this up such the way that they kind of go one and the other, vice versa. So this is going back and forth between this pixelated state to a non-pixelated state. Okay, so for the uh, epithelial to mesenchymal transition. So let me go to this one now, the MET first. All the cells start cytoplasm. Well, actually, I have another cell type that's called M, which stands for mesenchymal. Okay, so what I do is when my cells in a mesenchymal state, I convert out to cytoplasm state, and then I call the pixelization function that I had before. So that creates this uh, down way here that make all the cells to pixelize. Then every 2000 Monte Carlo cells has passed, I call the EMT steppable, where I go for all the cytoplasm of your cells. Okay, oops. Then I go over all the clusters. I collect all the cluster in a list, and then I create a mesenchymal cell. Okay, and then I convert all the pixels that I have inside the cell of all the compartments back into a mesenchymal cell, and I set its target and lambda volume. Okay, so basically I'm visiting all those pixels here and converting them, and I not really convert the pixel, I just kind of drawing the mesenchymal cell on top of all those pixels. Okay, and now this is going to become a mesenchymal cell. Does it make sense? Okay, so if you just, that's how I code it. And if you play this simulation, you should get something like this. And then they go, all the pixels get converted to the mesenchymal cell type, and then I convert them back again, and they should kind of sort out back one. And see that I'm not telling where the pixels should be, right? If you pay attention, right? Here, the first thing that I do is here. Try to get, yeah. That's the pixelization. That's what I do. I just draw all the pixels randomly inside the cells. And because I make a wise choice of the external contact energies, okay, they automatically sort out what it should be. The green cells like to be in the membrane because they're the basal compartment. The green cell, or the, no, I forgot the color, how the colors go. The other cell color, let's go towards the medium, right? And the gray compartments, they like to, which are lateral, they like to, they identify where a cell is and they go back to that cell. Okay, so everything else that happens here is just kind of guided by your external energies. Okay, and then later on, I convert all the pixels to a mesenchymal cell, and I chose mesenchymal cell property such a way that they like to spread along the basal membrane. Okay, so uh, and sometimes this kind of reclustering of the cell fails, and you have actually just two or one by the side. That can happen sometimes. So if I have extended this basal membrane to be all the way through, right? Maybe one of the cells is going to go too far away and it'll be harder than for them to go back. So that can happen. But I chose a basal membrane size to be small enough so that you can still see the cells spreading, but they can still find each other to go back to form the epithelium. So is it working in our system? Okay, what drives the selection with the cell type? So that is just imposed by me. Okay, so let me go back to the code that does that. It's here. So here I have a condition, my step function. I check if I'm not in the uh, if time is not zero, and every 2000 Monte Carlo steps, okay, I'm gonna do one transition. Okay, so this is the condition for me to undergo transition. Every 2000 Monte Carlo steps, okay, so that's what this is coding. I'm gonna either go mesenchymal to epithelial transition or epithelial to mesenchymal transition, right? So if my time is 2000, I get here, but 2000 is not divisible by 4000. Therefore, at 2000 Monte Carlo steps, I undergo a epithelial to mesenchymal transition. So I call this function here that makes all my cells to become mesenchymal, okay? Then time passes. When I get to 4000 Monte Carlo step, okay, I satisfy this condition again because 4000 is divided by 2000, okay? So I get into the transition, and then it is divided by 4000, therefore I call the MET one, which so I call this one where I convert all my mesenchymal cells, my dark yellow cells, into cytoplasm and then I pixelize it. So yes, it is forced. This is the clock that drives it. So it is uh, kind of uh, completely arbitrary, right? If you want to be this more realistic, then you can have some other condition that's gonna either call this one or call that one, okay? But the MIT and the EMT are already defined. Maybe you want to make your cells that are at the edge of your epithelium to to become mesenchymal. So you can substitute that by any other criteria that you want, and then you're gonna have a proper model for epithelial to mesenchymal transition. So the objective here was just to give another idea of how you can do this, and also to already give you some kind of code that you can that I'm happy for you to copy and reuse on your project if you want to. Okay. Okay, if not, let's proceed to the last exercise and the last example, okay, where we want to talk about Planar-cell-polarity, okay? So know that I think at least a few of you have seen the talk on Friday about modeling Planar-cell-polarity, and now we're going to do an exercise about that. So just a brief overview because most of you were not on that guest talk. Planar-cell-polarity is a very important and ubiquitous process of patterning that you see in development. Okay, so if you ever wonder if you just look at your skin right now on your arm, uh, chances are that all the hairs on your skin, right, are pointing the same direction. Is that true or not? They're all pointing the same direction. So somehow all those cells in your skin, okay, know that when they grow a hair, they should be pointing the same direction, okay? And that does not happen by accident. That happens because there was a pre-pattern mechanism along the epithelium of the skin that tells the cells to be oriented exactly in the orientation where the hair grows. The same thing happens for your hair here. The sensory hairs in your ear, okay, they have to be all aligned in a certain way. And actually, your whole cell, your whole body, right, they have to undergo some kind of conversion extension, like we have seen yesterday. And this conversion extension, when we did this exercise yesterday, we just impose some kind of polarity vector that you assume that all your cells already know how to rearrange themselves. And where does this rearrangement comes from? It comes from most likely come from this planar-cell-polarity patterning that happens in the cell. So first the tissue becomes polarized, and once it becomes polarized, it already knows exactly how to drive its movements. Okay, so that can be done by external morphogens, but it can also be done by direct cell-cell coupling the cells along the apical side of its epithelium. So it's called planar polarity because the cells are already polarized apical and basally, and so they form epithelium. But on top of that, on the apical side of the cell, you already have a secondary polarization mechanism that happens on the plane of the epithelium. So that's why it's called planar cell polarity. And what happens is that your cells have a group of proteins that is going to call that like to sort out across different sides of the cells and like to be coupled with the uh, other group of proteins across cell membranes. So you have this group of proteins called the Van Gogh, Prickle, again, funny names. Another one that's called is called Frizzle, Dishevel, and Diego. Okay, and they like to co-localize against the other group of cells across the cell membranes. Okay, so because they like to do that, they are initially in a mixed state, and they have such kind of affinity within themselves that where the green one goes, the red one goes to go on the opposite side. Okay, so that is some kind of ideal process that can be modeled. Use the concept of dynamic compartments. Okay, so that's what we'll be doing here. Okay, in this exercise, you're going to be doing this model to go about it. So for this one, I want you to download the file 7.2.4.zip, download the code, and please take a quick look at what's inside, okay? And you can even try to run simulation and tell me what you see, okay? So while you download the zip file, you're gonna see that you have four cell types. Again, you have a cytoplasm type, a P and D, which stands for proximal and distal, because usually when those proteins they colocalize, they define a proximal and distal axis, and the proximal is be colored as red, and the distal be colored as green to just match this picture that you have here from this paper. Okay, and you also have a lateral compartment just like you had on the epithelial example. Okay, and again, all the cells start in a C type and get pixelated. But on this example, all the external energies are set to 10 and all the internal energies are set to 0. Okay, so here we have to do a little bit of exercise to get the patterning that you want. So because all the external energies are 10, internal energies are 0, instead of getting a pattern, you get this kind of a bunch of beach balls in your epithelium and not this polarized state. Okay, so does that make sense? Did you get the file open and got a bunch of beach balls forming? It also looks like a Jackson Pollock painting, yes. Yeah, you can say that, but more rounded. Okay. So Nathaniel got it, Kyle got it, right? So yeah. Okay, you got it. If you want, you can print that and place on your on your living room. Okay. But you can also place the next at the end of the exercise output in your living room. Okay. All right. So you got this. So that's not what you want. What you want this to happen, right? The cells all start at pixelated state, but instead of getting this, you're getting the rounded Jackson Pollock output. Okay. But you want to have this one instead. So what we have to do? First, we have to start playing with the contact energies because unlike the previous exercise, I gave a really bad choice of energies for you to get any output. So, and again, like we did for the cell compartments example, we have to first make a qualitative choice for what the digital energies should be. So now I depend on your participation. Let's start with the contact internal energies. Uh, what they should be. So again, you don't have repeated circle parts in the cell, so you can ignore this uh, uh, part here. What you want these ones to be? They should be high, low, zero, negative. Tell me. Low for internal? Yes, it had to be low. We can say that maybe you want all of them to be zero or perhaps negative. Okay, actually a better choice to make it then all to be negative. So if you try that at your end, I suggest you to make all -2 instead of 0. I forgot to update my slides. But notice that there is one of them that I did not make that actually made positive, the proximal-distal. Why did I do that? I do that between the proximal-distal, the red and the green, because those two compartments they actually should segregate opposite sides of the cell. So yeah, I want to keep them apart. So I just make them to be have a medium uh, or a low or a positive value. Okay. Now, when I do things like this, I'm telling that in my model, the cells are able to self-polarize. Okay. And turns out that this is maybe true or not, depending on which system you have. So um, if you are cell in a fruit fly in a Drosophila, the cells can self-polarize by themselves. Okay, so you can so that's going to be a good model for Drosophila cells. However, if you're modeling mammalian cells like mice, it turns out that mice cells, skin cells, they cannot self-polarize. They remain in a mixed state. So a better model, if you are trying to use this to model mice morphogenesis or mice PCP or mammalian PCP, you should have all the energies to be negative because those cells cannot self-polarize. Okay, so this is kind of a fruit fly model, Drosophila model, because they can self-polarize according to the work of David Axelrod. But Danielle Davenport, she did the experiment in mouse epithelial cells, and they cannot self-polarize by themselves. So uh, then you have to model everything to be negative energies. Okay. Right, but anyway, we make our choice for the internal contact compartments. Now let's make external contacts. Okay. Uh, what is the most striking feature that we have is that we want to have this proximal distal coupling across cells. So what value should I use for the PD externally? Should it be negative, positive, high, or low? Any guess in the chat? Low. But it should be negative or should it be positive? Positive. Why positive? So yes, it should be low, but still should be positive because if you make that negative, then the two compartments are going to start to mix to themselves and you blur the boundary between them. Yes, exactly, Rita. Yes, Maisha. Yes, it doesn't want to go through the boundary. So you want to have low. So of course it has to be low compared to the other things, right? So let me ask you, what should be the cytoplasm energy be with the other cells proximal, distal, and lateral? If you want the cytoplasm to be inside the cell, you want those energies to be high, low, or very high? Yes, it should be high. So is exactly one of the things that we did on the first exercise. You want to be higher. So you want to be very high because you want the cytoplasm to be inside the cell and never see the other compartment from the neighboring cells. Okay, so what else? What about proximal, proximal, distal, distal? They should be high, low, negative. What they should be? Red, red, and green, green. Well, you do not want to have red, red coupling and green, green coupling, right? Therefore, the proximal, proximal, and distal, distal energy should be what? They should be high, right? So these also want to be high, maybe not as high as cytoplasm and everything else, but they should be high so we don't have red and red colocalization across cell membranes and don't have green, green colocalization across membranes. And now we can also talk about the lateral, okay? So the lateral should be more or less a neutral cell boundary, okay? So for that one, I just choose a medium cell contact energy. Does it make sense? So now, if this makes sense, what we did, we just made a qualitative reasoning about how those energies should look like compared to one another. And now we have to, once we made this kind of qualitative energy output matrix, we have to now translate that into numbers. Okay, so that will be left as an exercise for you. You can translate that to numbers by yourselves, and you probably want to make all these numbers to be negative. Okay, that I have zero. If you make negative, it's going to be better. But if you just make this kind of choice that I have here, then you get something that kind of looks like this. Okay, it's not as very as well as I show at the beginning, but you should get some kind of cell mix. If you wait long enough, you should get the whole tissue to polarize along one direction. So all my red are more to the left, and all my green are more to the right. Okay. Um, Arita, I cannot see very well on that one. Maybe you can maybe choose a higher value for the cytoplasm so they stay in the center. Uh, yeah, for now here, this is a complex model, so it takes a while until you get the uh, set of numbers that work. I also suggest all of you to turn off the cell boundaries drawing and only have the cluster boundaries to be drawn because that allows you to see the cells better. Okay. But if you just use these numbers, you should get something that looks like this because that's the output that I get with these numbers. Okay. All right. The next part of this, the final part of this exercise, these numbers should give some kind of decent patterning. Okay. Not perfect, but a decent patterning. But the last part of this exercise is create a vector field to so you can visualize um, the pattern that you get out of the simulation. But this has already been covered by Gilberto by the end of um, uh, last week, how to create a vector visualization field. I'm gonna, he has all the instructions on how to get that, because maybe just seeing this, you cannot clearly see that the cells are oriented, but if you make assign to each cell a vector that it has its tail towards the red and its point towards the green, then you can better visualize the alignment that arises from your model. Okay, so I'm gonna just stop here and hope that this exercise was helpful. So Maisha had a question and she herself read the answer. If the orientation was the fitting ellipse of the cell and plate the vector, you definitely can do that to see what's the going to be orientation of the cells. But in this case, it's just a vector connecting the proximal to the distal compartment of the cell as was defined here. Okay. Uh, yeah, so that's how it was defined. Okay. So you have all a copy of the slides available to you on the on the shared folder for the day. You can please, I invite you to do all this last part of the exercise. I think this had already been covered by Gilberto last week, how to do vector fields and also scalar fields. But and this is just so this is going to be just a refreshment exactly how to implement that specific vector field for this problem. Okay.